Hi, everybody. My name is Richard Border. I'm a new assistant professor in computational biology, and I'm going to tell you a little bit about my lab's research. So I'm a statistical geneticist, and I'm interested in developing methods for helping us understand how genes affect disease risk. There are a lot of reasons we want to know about this. First, if we know what genes are involved, we have a better picture of the causes and the biology underlying disease. And we can also hope to identify drug targets and do a better job at developing treatments. We can also learn about evolutionary context of disease and disease-relevant phenotypes. And finally, we can work towards personalized medicine, where we're able to optimize what treatments we present to which patients based on their genetic backgrounds. And so how do we learn about genetic architecture? How do we conceptualize this idea? Well, we're basically trying to learn a map from a bunch of features, the entire genome, and which is big, and any other non-heritable factors that influence disease, and then many disease phenotypes and disease-relevant phenotypes. This is a hard problem for a few reasons. One is that the genome is large. There are tens of millions of relatively common variants. To the extent that variants affect most traits, they tend to have very small effects, and many variants are involved. So pinning down exactly the relevance of any particular variant is quite challenging. We also don't know the functional form of this map between genetic and environmental features on the one hand and disease phenotypes on the other. So sort of how these factors combine. And finally, this isn't really a one-way map. Across multiple generations, disease phenotypes and disease-relevant phenotypes impact the joint distribution of causal variants and environmental factors through things like selection and mating processes. And because of all these things, as well as just sort of a fact of trying to do science, we have to sort of proceed in a somewhat two steps forward, one step back fashion, where a new technology or a new data type will come online. We'll come up with some new methods, get some promising results. The more time the community has to spend with these results, we'll sort of understand some challenges that are revealed, and then we'll try and synthesize what worked and what didn't, and come up with new, more robust approaches. Which brings me to sort of the problems I like to work on. So first, given the massive scale of genetic data, you have to be clever if you want to be able to do statistics on this data. And so a lot of the work I do consists of thinking of clever ways to sort of utilize math to make this computation faster, so we can actually make use of the data that we have. And particularly, I'm interested in using iterative and stochastic approaches to speeding up computation. I'm also interested in trying to make science as a whole more efficient. And so what do I mean by that? Well, as a methods developer, for me, science works in this kind of loop where based on existing results and you come up with a scientific hypothesis that you then formalize into a mathematical model so you can rigorously ask questions about parameters governing that hypothesis, right? Then, given this model, you have to come up with some way of doing statistics, right? So you have some data, you actually want to estimate these parameters. You also have to do some sensitivity analysis. And what this means is that, we have this mathematical model, but we probably don't have the right mathematical model, even if there is such a thing. And so we want to see that if we perturb the assumptions of our model, do we still get reasonable results? How do our results depend on these assumptions that we're making? And then finally, given that everything else is working well, we can actually do science, apply these methods we've developed, and hopefully learn something about disease biology. So there's a couple bottlenecks here. One is that it can take quite a while to go from a mathematical model to a statistical estimator that scales to the size of the data sets that we need to analyze genetic data. So I work on using distributed memory GPU frameworks to accelerate Bayesian inference with probabilistic programming languages to try and speed up this step. I also work on tools for making sensitivity analysis easier, more comprehensive, and more reproducible, trying to reproduce some of the recent advances that we've seen in machine learning due to commonly used benchmark data sets like ImageNet, et cetera, in the statistical genetics world. And finally, I'm interested in approaches that try to be robust to some of these classical assumptions we make about the relationship between genes and disease. And in particular, there's a lot of signal in the genome, but not all of it is relevant to the biological pathways between genetic polymorphism and disease. One way this can happen is through assortative mating. So our parents pass down genes to us, right? But our parents didn't select each other randomly, right? In fact, with respect to almost every phenotype or many phenotypes, mating is assortative, which means that our parents will be more similar to each other on a range of outcomes than two people picked at random from the population. To the extent that these phenotypes are heritable, that will over multiple generations induce long-range correlations between any causal variants for any of those phenotypes in the offspring. And genes aren't the only thing parents pass on. They'll also pass down environmental factors through the rearing environment. And so if we want to understand the impact of any given gene, we're going to have to account for the fact that the gene might also be tagging distal parts of the genome that also influence or that are related to the phenotype we're looking at through assortment, maybe not through biology or both, as well as environmental factors. And so that makes it particularly hard to figure out what any particular gene is doing. And if we fail to account for this, we can get quite misleading results. So one thing I'm really excited about is coming up with models that explicitly account for this dependence induced by these sort of dynamic phenomena like sortative mating, to come up with unbiased estimators that are able to parse this sort of population structure from the biologically informative variation that we actually care about. So hopefully you found this interesting, and thank you very much.